Greetings. This is a brief overview of the ESR facility. I am Gary Bettner speaking, and the technical director. I'm the director of the ESR facility, and the technical director who oversees the day-to-day operations is Mr. Brett Wagner. And the facility is located in the Eckstein Medical Research Building down in the basement, room 64 and 68. Front door is room 64. It's right by the drinking fountain and restrooms and west elevator. So in the ESR facility, we always begin with two simple questions. Well, they're never simple. What is your scientific question that brings you to the facility, and what types of samples are available? And after we discuss, brainstorm, whatever is appropriate, then we decide on what service might be employed, depending on your goals. What can we offer? Well, EPR spectroscopy. ESR is electron spin resonance, EPR electron paramagnetic resonance. There are two different names for the exact same spectroscopy, no difference. So we flip back and forth in our vocabulary here. So what is EPR spectroscopy? It's a spectroscopy where we can look directly at free radicals, or sometimes we have to use spin trapping techniques because some of the radicals we want to study are just too short-lived and at too low concentrations to see. We can look at redox active transition metals, for example, iron 3, cobalt 2, copper 2, cobalt, vanadium. There's a whole host of metals that we can see. We have a Clark electrode where we can detect oxygen uptake by biochemical, and cellular systems. And we have a Seahorse XF96 where we can look at rates of oxygen consumption and extracellular acidification to look at how mitochondria or whole cells function in metabolism, how they make ATP. We have a Sievers NOA system to detect the footprints of nitric oxide. Keys are examples are nitrite and nitrate. And then we have many quantitative assays to look at small molecule antioxidants in other species, for example, vitamin C, superoxide, GPX4, etc. And then we have some assays using redox active dyes, or we can give advice on how to use them using techniques in your lab or other facilities, etc. And there's many more things, so just ask. I'll go through each one in just a very brief example of each. So this is one of our EPR spectrometers. We have two of them. They look almost the same. There's just a very minor difference in their configurations, and we can go back and forth between instruments depending on what experiment we want to do, or we can do the same experiment on the different instruments and the results are absolutely identical. So EPR spectroscopy, we can detect free radicals directly. Examples are like vitamin C radical. I show you the spectrum here, very simple spectrum. In EPR, the spectra you see are always the first derivative of the absorption curve. That's why they go up and down, etc. We can see semiquinone radicals, nitroxides, or just a few that we can see directly, but very short-lived radicals such as superoxide, thiol radicals, carbon-centered radicals, etc. We usually are not able to see directly, and so we use spin trapping. And here's an example of the superoxide spin adduct, as it's called, with the DMPO spin trap. Okay, EPR spectroscopy of metals. So here are some examples. On the left, you see this is an example of whole blood. And here we can see the ceruloplasmin. It's a stress protein in the blood. And here is a standard of ceruloplasmin, so we can see that they behave identically. Here is a small signal of nitric oxide on hemoglobin. And then here's a different kind of signal of from the same sample, all different instrument settings. And here we see methemoglobin over here, this little bump here, and that can grow or decrease. And here we see transparent, all in the same kinds of samples. On the right, we see where we coordinated iron. This is iron that's coming out of ferritin, not in the ferritin, with desferrioxamine or desferrial, and we get this kind of signal, okay, for the iron. And here is the same species, but here we captured the iron from tissue, and we were able to quantitate how much labile iron in tissues. This has to be a tumor homogeneous. Okay. So there are many examples. Okay. Here's our Clark electrode. It's a very simple instrument. We have a A to D converter and something to control the voltages, shall we say, on potentials on the electrodes. And here, you know, if things are being oxidized, oxygen disappears. Simple as that. Okay. And so we look at the change in concentration of oxygen with time. And here are different hydroquinones, and this represents their rate of oxidation. Okay, and it all depends on what's on the rings. You see OH, etc. And then we make a plot. Here we made a plot of the log of rate versus fraction ionized, and we get basically a straight line. And all this from the simple oxygen consumption data, and we were able to gain a lot of information about the free radical out of this indirectly and the pK of the system. On the screen itself is oxygen consumption when we have a cell suspension. And we see basically a line going down here. So it's zero order in oxygen as we see. But if you really want to do oxygen consumption from cells, we should use the Seahorse XF96 in the facility. And with this instrument, we can collect two kinds of data: oxygen consumption rate and extracellular pH changes in the media. So OCR is going to reflect oxidative phosphorylation, and the pH change in the media, providing you do the proper calculations, can reflect the glycolytic production of H+, and from that we can get the rate of ATP production in cells. Here's the example, the special Seahorse plate where you seed your cells. The outside rows are for standards, etc. And this goes into the instrument for the measurement. And these are example data. This is a classic kind of data that you'll see published in many papers in the literature. And this is the oxygen consumption rate, okay, basal rate, and then we add oligomycin to determine how much ATP is being used for ATP, etc. And over here, we have, from the same kind of experiment, we have the extracellular acidification rate. That is the rate of change in pH versus time. And we see a background level, and then when we add oligomycin, it goes up a little bit, etc. And what we do is we use the number of cells present, the volume, and all kinds of information that we collect when we do our experiments, so we can quantitatively convert this to a proton production rate on a per cell basis, and then a proton extracellular rate, the PER as it's called. And with these two kinds of information, when we put it together, we can get the rate of ATP production, okay, by the cells, and we can dissect it. We see how much comes from respiration and how much from glycolysis. We can use different substrates, et cetera, so we can really dissect on a absolute quantitative basis the flux of ATP in cells and where it's coming from. So our Sievers NOA, excuse me. What it is this is the basic instrument. You know, we have a computer with it, obviously, to collect the data. And this is a reaction system right here. And so we can, with appropriate standards, we can determine the nitric oxide concentration in solutions. We use this if we're making standard NO solutions. The big use is to determine the total NOx. And many people want to dissect that to figure out how much nitrate and how much nitrite there are in the samples. Nitrate is usually what we concentrate on when we have blood plasma or serum, etc. Nitrite is typically what we concentrate on with cell culture medium. And so here we see, you know, in an organism, you know, rat, mouse, people, the nitrate levels in their blood. And this is a measure production of nitric oxide in your cell culture system. And there are many other possible samples. They need not be from cells or whatever. They can be water pollution samples, whatever. We can do it all that way. We also have quantitative assays for a wide range of species. So we have a microvolume spectrophotometer, and with that, and as well as a plate reader, assays that we've developed in our lab to quantitate vitamin C, you know, anything from orange juice to blood. We have an assay, a combined assay that we just developed to measure GPX4 and GPX1 from the same sample at the same in the same run. Is absolute quantitative, and it works very well, saving a lot of time and effort for those kinds of experiments. We can give absolute quantitation of catalase and SOD using our quantitative redox biology techniques. We can provide you information on intracellular concentration of H2O2 via an inhibition of catalase assay that we use in our lab. And we can detect superoxide fluxes using UV visible spectrometry, EPR, HPLC. And one of the big things in our lab is iron. We can quantitate iron in certain situations, but mostly the control of iron and the removal of iron from buffers, etc. We can give advice on that because iron plays a role in so many things. For example, in redox active dyes. In the typical redox active dyes, such as DCFH, as people usually shortcut name for this, iron is absolutely essential in causing the oxidation of the dye, so the color change that people see. And so we have to really be careful how we interpret data. We have this HPLC system that you see pictured here, where we can quantitate superoxide from DHE and mitosox, okay, etc. But we can also see things that people usually do not see when they're just monitoring fluorescence and really understand what's going on with these dyes and what might be going on to the system. So is superoxide that you're allegedly seeing in other assays, is it real or is it artifact? Those are the real questions, okay? So I just want to remind you, iron, a little bit of iron can make a big change in your results when you do redox biology. So in the ESR facility, our goal is always to do some good. And if you have any questions, just ask. Contact Brett or myself. And here are some websites. They may not be all up to date. We're going to be working on that this summer later. The ESR facility is part of the Radiation and Free Radical Research Corps in Holden Comprehensive Cancer Center. We report to a lot of people. You saw those logos on slide 1. A lot of people looking over our shoulders administratively. But anything we can do to help, we would love to. So thank you very much for your attention. And I will